Hello and welcome everyone to our final installment of the ISCN 2024 Updates series. Today's talk is on optical genome mapping, and it's by Drs. Jean McGowan-Jordan and Dr. Hastings. Dr. Hastings is a state-registered clinical scientist and a fellow of the Royal College of Pathologists. She has more than 35 years experience in constitutional, acquired, and molecular cytogenomics, both in a research and diagnostic setting in the UK. Dr. Hastings is the chair of the ISCN Standards Committee since 2020 and editor of the ISCN 2020 and lead editor of the ISCN 2024. And we thank Dr. Hastings for her previous two talks. And joining her today is Dr. Jean McGowan-Jordan. Dr. Jean McGowan-Jordan has led the CEHEO Genetics Diagnostics Lab. She is a professor in the Department of Pathology and Laboratory Medicine at the University of Ottawa. Her lab has introduced a standalone clinical test for facioscapulohumeral muscular dystrophy using optical genome mapping and is implementing this exciting technology for analysis of acute leukemias and myeloid disorders. Dr. McGowan-Jordan has served on the CCMG Board of Directors, the CCMG Genetics and Genome Diagnostics Working Group, and the Cytogenetics Committee. Internationally, Dr. McGowan-Jordan was the chair of the Standing Committee for the ISCN from 2013 to 2020, serving as lead editor of ISCN 2016 and 2020. She is currently an advisor to the group and co-authored the new nomenclature for the genome mapping in the ISCN 2024. Please remember to place all questions in the Q&A. They will be answered at the end of the session. And now I will turn it over to Dr. Hastings and Dr. McGowan-Jordan for their presentation on ISCN 2024 updates for optical genome mapping. Thank you.
Thank you, Celeste. I am now going to share the talk. This hopefully will be slightly shorter than the other talks, but we will see. See that now. So Jean and myself are going to talk about the optical genome mapping, and this is relevant to ISCN 2024. It is new to ISCN 2024. There was a paper published in the beginning of last year on optical genome mapping, and this is an update on that. So what we're going to cover is before I actually say what I'm going to cover, sorry, let me just talk about a potential conflict of interest. I am the ISCN standing chair. I'm also a consultant for GenQA, which is Genomics Quality Assessment Provider, which is a global ISO accredited EQA provider and part of the UK NEQAS consortium. We do offer EQAs and composite tools for ISCN, but being as we support ISCN, there technically isn't any conflict of interest. Oh, just for information, EQA in the UK is what we refer to as external quality assessment, and you refer to it as proficiency testing in the States. If in the ISCN you see some asterisks, please do not think they're part of the ISCN. They are not. It just denotes ISCN examples in ISCN 2024, which are shown here with permission from Cargo, who's got the copyright of the ISCN. So we're going to talk about the optical genome mapping. I'm just going to give you some basic background on the nomenclature. Jean is going to go into a lot more detail and give you specific examples, and then we've got a short quiz for you to see if you can apply the rules. So if you haven't already bought the book, or you haven't gone online and purchased just an online version, please do so, because the ISCN 2024 is totally different from our previous versions. There's been a lot of changes, and it's been amalgamated and reviewed to make it easier for you to use as diagnostic departments. So please do get your own copy. If you do buy the book, you get the online version for the whole period for which the book is available. So all the ISCN errors will be corrected on the online version, and you will. That's another reason for getting hold of the online version. So the chapter structure is such that in ISCN 2020, there were 20 chapters. Now there are only 16. And the ones in red are where we have made some changes, considerable changes. There are a lot more examples across every chapter. But the historical introduction has been introduced, has been condensed. The normal chromosomes now incorporates chapter 7, which was on variants. The general rules includes many chapters, and that gives guidance on the general rules for ISCN. The karyotype incorporates part of chapter 8 and chapter 9. Then we have a specific chapter 9 on genome mapping, which is what we're going to discuss today. And the chapter 10 on region-specific assays, including targeted analysis, has also been expanded from previous versions of ISCN. And if you are looking for the ideograms, you'll find them in the appendix. We moved them. So just to explain the ISCN formats in genome mapping, it will depend on what information you're getting from the technique and the technique obviously that's used. So if you know the structural information from the assays, then you can use the karyotype format. If you cannot get the structural information, then you use the microarray format. Now, the karyotype format comes in three ways, which is abbreviated, short, and detailed. And the microarray has abbreviated, short, and extended. And they're very similar to array and in the way that we use the technology, use the nomenclature and RSA. So the abbreviated format in karyotype, the first example is just for a single X chromosome, and you just put OGM square brackets ×1. In the short form, then you are talking about the actual abnormality. And so because it's karyotype format, you put both breakpoints, even if it's the same breakpoint for the deletion of 22, and then you put the nucleotides, but you do not put a copy number at the end of that. And then for the detailed format, then you're describing it from Peter to Cuta, and you're doing that both in the karyotype format at the very beginning here, and then following that same process to describe the nucleotides. With the microarray format, then it's very, very similar to the microarrays. In fact, for a triploidy, you'd put this, 3 copies of X, 1 to 22. You will put the copy number in for the short form, and for the extended, you're putting the flanking normal nucleotides that were seen on the software. Commas are optional in the ISCN, except for the extended system, because if you start putting commas in here, it becomes confusing if you're talking about 2,188,000, or it's ×2 and then 1,885,000 or whatever. Another thing that you can do now is that you can tabulate, well, you always could, but we've given you some examples so you can tabulate your complex results, because if you're dealing with neoplasia, often you are dealing with complex results. And even in constitutional, we're finding things aren't as straightforward as we used to think they were. And this can be done for both array, OGM, and sequencing. And we've made some suggestions about what information should be given. And so we've suggested that you should include the chromosome band, the genome build, genome coordinates, the abnormality, the type of abnormality, loss, gain, amplification, region of homozygosity, the proportion of the sample of the abnormality where it's applicable. And also in the comments, you could put any genes that were particularly relevant. Obviously, if you've got 50 odd genes, you're not going to list all those genes. We're just talking about, say, in a neoplasia, where there's a specific gene that is relevant to the disease, or in a constitutional situation where you've got a particular syndrome and there's certain gene or genes that are really relevant to that. So just to give you a bit of background about the genome mapping and how it all came about, we started discussing this as a group way back in 2021, and we only had 3 examples at that point because it was a relatively new technology. By August in 2023, we had 81 examples, and that is mostly due to the genomic mapping community, who really helped in establishing this nomenclature. So the committee came up with ideas, it went back to the genome mapping community, and they fed back where they thought it would work or ways in which we could improve it. So it's been a really good interaction between the committee and the genomic mapping community. And without them, we couldn't have done this nomenclature or got it so far advanced for the first time it appears in print. And it has been published in 2023. But there have been some updates since then, so you'd be better off looking at the ISCN, to be perfectly honest, the book itself or online. So normals are very straightforward. It's identical to what you'd use in arrays, and the normal aneuploidy genome mapping nomenclature, as I say, it's like RSA and array nomenclature, and it's exactly the same whether you do constitutional or neoplasia. Also, if you know you've got structural aberrations, then you will put in the deletion, duplication, insertion, inversion, translocation, complex, depending on whether you're using karyotype or microarray format, because in some cases you won't actually know the structural information. So you won't know it, say, for a translocation sometimes, but if you did know it was a translocation, then you'd use the karyotype format. So just to explain the karyotype and the microarray formats in a bit more detail, as I mentioned before, the karyotype format is only used if the structural nature of the abnormality is apparent, but no copy number is included. So for the abbreviated form, in the microarray format, it's used for aneuploidy and chromatogenesis. Only the chromosome is given, no nucleotides, and the copy number is given for aneuploidy, but it isn't given for chromatogenesis. When you use the short system in the karyotype format, you define the structural abnormality by the chromosome breakpoints and nucleotides, but there's no copy number. But if you use the microarray format, you define the abnormality by the chromosome breakpoints and nucleotides, just the same as in the karyotype format, but you do give the copy number because you're not giving the structural information. And in the detailed form, you're describing the abnormal chromosomes in their entirety from Peter to Cuta. And for a derivative chromosome, it's not necessary to use the terms del, do, inversion or translocation, because the structure is apparent from the nucleotide sequence, but you again do not include the copy number. Whereas for the extended system, in the microarray format, you include the span of nucleotides involved in the abnormality, as well as those that are flanking uninvolved nucleotides. And commas are not used in this particular format, but copy number is given. So when you come to genome mapping, the platform, labeling strategy, bioinformatics pipeline are all indicated in the narrative. They are not given in ISCN. If you've got multiple abnormalities, these could be presented in a table if you wish. They don't have to be. Only the clinically relevant abnormal results are given. Normal sex chromosomes may be reported for the purpose of sex determination. So if you've got gain of an X chromosome, you might want to know if it's on a female background or a male background. If you've got deletion of an X, you might want to again know it's on a male background or a female background. So you report the normal X or Y chromosome in those situations. And again, following all the rules we've had previously, the aberrations are listed in numerical order from the lowest to the highest numbered chromosome, where the sex chromosome abnormalities are always listed first and then the autosomes. Band designations and aberrant nucleotides are listed from Peter to Cuta. Multiple changes of homologous chromosomes are presented with the abnormality closest to Peter being described first. And we have, after much discussion, it was agreed that we would include the variant allele frequency in the ISCN. But the committee is also aware that the nucleotides may indicate the extent of the fragment determined by the positions of the labeled DNA motif within the variant region and not necessarily those of the actual breakpoints, but that's just the limitation of the test. And like we have with all other techniques, there are limitations and we have to be aware of them. I'm now going to hand over to Jean, who is going to talk about the more specific optical genome mapping. I will forward the slides on for you, Jean, if you just say next slide, and I will move on.
Perfect. Thank you so much, Roz. And thank you everybody for joining in. I'm going to go through some different structural abnormalities and numerical abnormalities with you, just to give you a flavor of how the things that Roz has explained, how they're applied in actual examples. And just to go back to Roz's thought on or point about the nucleotides that are listed is the position of that DNA motif that's targeted by, in many cases, the optical genome mapping platform that most of us are using. And it's similar to microarray in that you can only list the position of the probe that you have on your microarray platform. So it's very analogous to that, if you want to think about it that way. So here we have examples of a deletion on chromosome 22 on the long arm. And so in the examples that are given, you'll see, as Roz mentioned, there are two different ways of showing a deletion, whether you want to describe it as a copy number change or whether you want to describe it with the structure of that deleted 22. So the first two lines there are the microarray-like format, where you have the copy number times 1, and the extent of the nucleotides where the DNA motifs lie, that you know that copy number change has occurred. And you only list the breakpoint once, just like our microarray format, so 22q11.21. The second line is microarray format as well, but it is the extended format. So you have those flanking normal nucleotides listed in your nomenclature. And then the third line is more like the karyotype. So you're describing the deleted chromosome 22. And so you repeat that breakpoint because you're giving both the proximal and distal breakpoints of the deletion. And then you follow that with the nucleotides of that region that you know is deleted by the position of those DNA motifs. So and again, the karyotype format is given without copy number because you're describing the structure of that deleted chromosome 22. If you have something like a mosaic, this example is a mosaic loss in a patient who has constitutional trisomy 21, for instance. So this is a deletion on the long arm of chromosome 17, given in array format. So the extent of the deletion there, and it affects the proportion is in 50% of the sample. So it's an acquired abnormal or a mosaic loss. And the constitutional trisomy 21 is indicated by using C, just like we do for all other types of nomenclature that we use. C means constitutional, and that is 100% of the sample. So 1.0 is added for clarity. Okay, we can go to the next slide.
All right, there you are.
Excellent. Okay, so here are some more examples of deletion, this time more in a neoplasia type setting. So we can, again, this is a deletion on the long arm of chromosome 17. So you can describe that in array format in the top two examples. And if there is a gene of interest, such as NF1, that is, clinically significant, we decided that it would be valuable to have that indicated as part of the nomenclature. So you have that option to add the gene of interest after the brackets with the nucleotide numbers. Similarly, if you're using the karyotype format to describe that deleted chromosome 17, you can have it with or without that gene of interest. And here is a constitutional mosaic in a neoplasia sample. So we have a deletion or a copy number loss of this region on chromosome 17 on the long arm, and it's a constitutional mosaic, so it's a loss in 50% of the sample. Or you can describe it as a deleted chromosome 17 using the karyotype format. You can mention the gene of interest. So this patient would be constitutionally deleted for NF1, and that's indicated by including NF1 in the brackets and then following that by C, indicating it's a constitutional loss. Okay, next slide. For clarity's sake, when we're dealing with sex chromosomes, always helpful, I think, to include the normal sex chromosomes. So this is a bit of an exception to our general practice where we only describe abnormal chromosomes. But when you're dealing with sex chromosomes, for clarity's sake, it's often helpful to include the normal sex chromosome. So in this way, you know, it's an XY individual and the single X chromosome has a copy number loss. So it takes it down to 0 for that region at XQ25 that is missing. So again, you can do that either by the using the microarray format or the karyotype format. They are equally valid. If it's an inherited abnormality, you can indicate that, again, just like we do for other types of nomenclature for different technologies, by using the abbreviations MAT or PAT or INH. And so just place that after either your copy number or after, if you're using the karyotype format, after those brackets with the nucleotide numbers indicated. Okay, next slide. All right, duplications, as we know, are always a little more challenging. And so this format, this slide has a lot more writing on it. So we have developed a system of using, describing duplications using the short system. Again, similar to microarray in that top red line, where you have a copy number of 3 indicating copy number gain, or you can use the karyotype format listing the cytobands Q21, Q25 indicates that that duplicated region is actually in the same form, is in the same orientation relative P-tier to Q-tier as the normal. But if you want to give more detailed information about orientation, which is important, especially if you have an inverted duplication that can have implications, you can use the detailed format where you basically draw out the chromosome. So if you had the orientation that was maintained, maintained relative P-tier to Q-tier, it would be as outlined in the, where it says duplication detailed system orientation maintained. And if you walk through that, you'll see how it follows P-tier to Q-tier. But if you have an inverted duplication, depending on where that inverted duplicated segment lies relative to the normal orientation fragment, that will change. So this describing the derivative chromosome 6 from P-tier to Q-tier, and then in the second set of brackets, drawing it out with P-tier and then the nucleotides, and using the double colon to indicate those fusion points throughout this type of detailed system. I think that's good. Okay, thank you. Okay, now we're on to translocations. So translocations, because it's a structural abnormality, you're not going to use a microarray format. You're going to use the karyotype format because you're describing the structure. And that's the beauty of optical genome mapping. It has both the benefits of the benefits of G-banded karyotyping in that you can find structural abnormalities and copy number changes. So that's why we had to have these options for the karyotype or the microarray format. But for structural abnormalities, you're always going to be using the karyotype-like format. So one thing I didn't mention is that anytime you have nucleotides in your nomenclature string, you have to include that genome build after the acronym OGM, just like microarray or indeed RSA. We always include the genome build so that gives context to those nucleotide coordinates. So here we're just describing a balanced translocation between chromosome 6 long arm and chromosome 7 long arm. You'll notice here that there's a tilde. So, and that's to indicate sort of an imprecision or some question about where in that region the breakpoints occur. So anytime you have uncertainty, you can use the tilde to indicate that. So, and going back to the string, so it's translocation 6, 7 with the cytoband breakpoints and then the nucleotide breakpoints. The only difference here is that we've included an example that had uncertainty, so we've included the tilde. If there's a fusion that's clinically important, as we come across a lot when we're doing neoplasia samples, we have the option to include that fusion gene in the nomenclature string. So here's an example of a translocation 312. And again, the nucleotides are given with some uncertainty, but we know that this creates a MECOM ETV6 fusion with a variant allele frequency of 0.3. So that's that in that order. So cytobands, nucleotides, fusion gene, and then the variant allele frequency is how that's presented. So I think that, clinically that's very important. And you'll notice that we're using the accepted, I think it's HGBS or Hugo format for describing fusion genes with the double colon between the genes of interest. Okay, next slide. This is an example of complicated, but things we come across fairly regularly when we're doing these types of analyses. Three-way translocation that produces a fusion gene. So this is a 6, 12, 21 translocation. Again, listing the cytobands that are involved in those and the translocation breakpoints. The nucleotides all have some uncertainty in this example. But importantly, this translocation has been shown to produce the ETV6 RUNX1 fusion gene. And in this example, again, then the variant allele frequency of 0.4 is listed at the end. Okay, next slide. Thanks. Unbalanced translocations. So we know the structure, so we're going to use the karyotype-like format. This is an example of a supernumerary chromosome. It's an additional chromosome 21 that's derived, or 22, it's derived from chromosome 22, but it's from a 1122 translocation, that recurrent constitutional translocation, with the breakpoints listed there and then the nucleotides. We're going to indicate that it is derived from a maternal translocation by using the short form DMAT, so derived from maternal, just like we would in when we're describing G-banded chromosomes. So it's very, very similar. The only thing different here is that we're including the nucleotides. And of course, we have to use the plus to indicate that it's a supernumerary chromosome. Okay, all right. Now, this is where it gets a little bit more exciting and challenging. Inversions. So inversions that are within a chromosome can be really difficult to, if not impossible, actually, to discern from a translocation between homologs. So the committee and the genome mapping community came up with specific nomenclature. And this is because the mechanism really isn't apparent using genome mapping, because it doesn't allow the technology doesn't allow the homologous chromosome 16s in this example to be distinguished from each other. So here we have a well-known abnormality in version 16. So you can present it either as inversion 16 PQ with the nucleotides, or translocation 1616 with the nucleotides, and then that creates the CBF beta MYH11 fusion gene. So in this, if you use that format, you'd have to include "or" in your nomenclature, because you really can't distinguish between those two possibilities. So you don't know if it's an inversion 16 or a translocation, because by genome mapping, they look the same. What you're seeing is those fragments that create that fusion gene. Alternatively, you can report the ISCN without giving the mechanism. So you can just say that there's a fusion 1616 with those breakpoints and those nucleotides, and that it creates the CBF beta MYH11 fusion. So that's an option. So it's either this long one giving the two different structures, or just giving the information about the fusion 1616, which is really the clinically important information. Okay, we have nomenclature to describe interchromosomal insertions. So in the top example, we have an insertion of chromosome 13 material, long arm, into the long arm of chromosome 18. So very similar to, again, its structure, so you're going to use the karyotype-like format. And so we have the nucleotides with some uncertainty on chromosome 18, where that insertion occurs. And then the span of the nucleotides from chromosome 13 that are inserted into that. You'll notice that the nucleotides on chromosome 13, we have inverted in orientation. So that means that fragment from chromosome 13 is inverted relative p-tier to q-tier into that chromosome 18. So because you're starting at 77 million and then going down to 35 million. Just like karyotype, G-banded chromosome karyotype, we use the semicolon to distinguish between the breakpoints and the nucleotides from those two different involved chromosomes. In the second example there, we have a derivative 2 from an insertion of something we don't know, that's indicated by the query, into band Q25. So all we know is that there's some extra material, but we don't know the origin of that material, so we can use the query. So it's a derivative chromosome 2, and then we use a semicolon, the query, the nucleotides on chromosome 2. Again, there's some uncertainty about exactly where that insertion happens, and then semicolon query. Okay, next slide. Some other examples of intrachromosomal insertion. So this is a balanced example where we have within a chromosome 2, we have insertion of material from the short arm further up into the, more distally, I guess, into the short arm of chromosome 2. So from P22 to P21, that fragment is inserted into chromosome P24.1. So again, the insertion point is indicated at the beginning of those last brackets, but there's some uncertainty. It's 19,700,000 and something. The breakpoint, or the fusion, is indicated by the double colon, and then we have that 7 megabase region roughly from more proximal part of chromosome 2p inserted further up. So that's how the intrachromosomal insertion nomenclature works. If you're going to want to describe it in the more extended version, which can be useful, you know, because it really draws out the chromosome from P ter to Q ter. You follow a very similar way we would do it to in G banding. So you'd say derivative 2, and then start at the P ter, say where the breakpoints are, use the double colon to indicate those fusion points, and then keep adding to it till you get to Q ter. And then you would explain more in terms of nucleotides. So you start at P ter and then give your the extent of the region with the ending nucleotides, the fusion breakpoint again with the double colon, the nucleotides that that flank or are the extent of that inserted region, and then show how the chromosome continues until the Q ter. Again, an example where the tilde is used to demonstrate those regions of uncertainty. Okay, next slide. There is nomenclature that's specific for ring chromosomes. And again, it's structure, so we're using like a karyotype-like format. So we have a ring chromosome. Of this example of chromosome 8, where you have breakpoints at P2 3.1 and Q2 4.3. The breakpoints in the nucleotides, there's fusion between the 9 million and the 144 million. So you have the breakpoint at the P term, the nucleotide, that's the first normal one, going all the way to the Q2 4.3 breakpoint, which is 141 million something. And then indicating that that's fused with the double colon again. If you have uncertain breakpoints, you would just use the tilde to indicate that, as in this example. Yeah, and notice that, you know, depending on what software you're going to be using, this can be also detected both as a fusion and perhaps as a deletion as well, if those regions that are lost at the termini of the chromosome are detected as a copy number loss. Okay. I think this is probably the last slide on the specific examples, but I just wanted to let you know that there are in this section 9.4.2.7 or 0.2, I guess, there are examples of how to describe complex genomes, because as Roz mentioned, you know, we're finding things that are much more complicated by genome mapping than are apparent by our previously used technologies. So in this figure 13, we had this example actually provided to us. It's a complex rearrangement between chromosomes 3, 6, and 18. And it's a rearrangement with duplication within band 18q2 2.1. And so you'll see I'm not showing it here, but in the actual nomenclature that we painstakingly worked through, it describes each of the individual derivative chromosomes. Because you can see these very complicated things, and sometimes we really can't resolve them, we have examples to use, whether you think it's a chromothripsis type mechanism, chromoanasynthesis, or chromoplexy. And so, you know, chromoanasynthesis generally is confined to one chromosome. So here you can use a short version and just say OGM 17 CHA, or you can give the extent of the cytobands and the nucleotide positions that are involved in those chromoanasynthesis events. Or if you have something like chromothripsis or chromoplexy, you give the chromosome bands, or so chromosomes or the chromosome arms that are involved, and then use either CTH or CPX, depending on what it looks like is the most likely mechanism. Okay, I think that is Next slide.
that's I'm on to the next one. Just for information, this 3618 rearrangement was actually a constitutional case. So that's why I say we're finding out more things all the time. So I'm just going to briefly talk about some educational tools which might help you, and then we'll go on to the quiz. So obviously you can go into the book, you can go online. There is a forum online which could be utilised much more, and you can put your queries and ask questions, and if you pick up errors or anything, you can put that on the forum as well, and we will respond as a standing committee. Not that rapidly, I'll be honest with you, but we will respond. There also are external quality assessment and proficiency testing providers. There are many national and international providers that include ISCN in their assessment, so you can check that you're getting your nomenclature correct. Some have specific genome mapping EQAs. The one I'm involved in does, as far as I'm aware, they're the only ones currently doing this. And there are publications. So a summary of all the ISCN changes has just been published in Cytogenetic and Genome Research last month, or might have been 2 months ago. There are also individual composite tools. There's a modular one provided by GenQA called Genie, and it's done for ISCN, and genome mapping will be included in that. It's not released yet, but it will be, as is HGBS. Currently it's basic level, but we will add an intermediate and an advanced level as well. But if you want to just get more familiar, there are webinars. The ASHG has one from 2 years ago, but it has been, there have been some changes since then. This summer in August, we're going to do some complex cases with ISCN for you to work, for people to work through, so you can use that. There are some videos on GenQA YouTube. They're currently ISCN 2020, but they will be updated to 2024. And there are obviously workshops. So GenQA offers workshops, and of course you can go back and look at the CGC seminar series of 3 seminars to revise, if you like, or refresh your memory about how to use ISCN. So we're now going to go to the quiz. So this will come up as a poll. So you'll need to look at the question and then submit your answer, and then we'll go through the answers. So the question one is, I hope, is easy for you. And that's a normal female result using genome mapping. What's the correct ISCN 2024 for this result?
I went ahead and shared the result.
Thank you. We'll close that and move on. So I'm pleased to say that the largest proportion got the correct result. The first one is incorrect because you need to put the X and the 1 and 22 because they've got the same copy number. They're all put in one bracket. And there also needs to be a space after the OGM. And that's something that's new again to ISCN 2024. This B needs a space after the OGM, and 23% thought that was the correct answer. And this one, again, it's the same copy number, so it does need to be in one bracket, even though the space is given there. So the second question is, this is an interstitial deletion of 17q11.2. So what's the correct ISCN 2024 for this result?
Okay, shared.
Thank you. And again, the vast majority of you got it right with 41%. And it is the first answer. You could alternatively write it in the karyotype format if you definitely knew that it was the structural rearrangement without copy number. So if you do use the karyotype-like format, you have to put the breakpoints in twice. And so that's why about 27% of you made that error. With this one, this is the, close that down. This is array like ISCN. So you should only put the breakpoint once, and you need a ×1 here. Only a small proportion of you thought that was correct. And then this one, again, it is the karyotype like, and therefore you should put breakpoints in twice, and there is no ×1. Next question. So this is a balanced translocation. Again, with OGM, you can pick up balanced results of a 312 that results in a fusion between MECOM and ETV6. Now, my question is, which is the correct ISCN for this result? And assume there's no uncertainty in the breakpoints. It was just so we could fit them on one line. It wasn't too difficult for you to read it. We took the uncertainty out.
Shared.
Thank you. So here, you seem much more competent on this. This is brilliant. So only one person thought this was correct. And that's just that the MECON, and there's also a bracket missing, and the ETV6 should be described at the end of the ISCN string. 67% of you got the correct answer, very well done. 17% thought this was the correct, but the breakpoints are missing for the 312, and because you're using the karyotype format, you do have to put the breakpoints in. And this one is wrong because you're giving nucleotides, so you do need the genome build, and 14% thought that was correct. So it's just little reminders of the things you need to look out for. Next question. So you've got a ring X8, that has been identified by molecular genotyping. So which would be the correct ISCN for this one?
Shared.
Thank you. So 66% think the first answer is correct. So let me show you what the answer is, which again, so the structural rearrangements you seem much more confident in, which is great. So yes, A is correct. B is not correct. 70% thought it was, 17% thought this was correct because you're talking about structural rearrangement, you've got a ring X, so therefore you don't put ×1. afterwards. If you know what structural rearrangement is, you don't put ×1. C is incorrect if you just thought that was correct, and that's because you're missing the fusion, break and fusion, double colon before and after the nucleotides to show that you've got break and join, and also you wouldn't include the ×1 because you've got a structural rearrangement. And again, this one, the last one, 10% thought was incorrect. You're still missing the fusion and join double colon, although you haven't got the ×1, which is good. Now this is the last one. So this is an additional DIR22 that's identified using genome mapping, and the mother carries a, she carries a balanced 1122. So this is in a child.
Shared.
Thank you. You're getting a lot more confident, all of you. This is brilliant. So 73% this time got the correct answer. So we're seeing improvement as we're going through the quiz, which is fantastic. The first one is incorrect. It's an easy mistake to make. 15% of you made it. And that is because it is an unbalanced derivative from a balanced rearrangement, it becomes DMAT, not MAT. Because if it was MAT, it means mother carries that same DIR22 and she doesn't have the DIR11. C, 8% thought was correct. This is a very unusual situation whereby in the genome mapping we have a plus, because otherwise you don't know you've got an additional copy, and you're using the karyotype format, so it's the only time you would use the plus when you've got this additional chromosome. And you can't use it because you're using the karyotype format. You can't do copy number. And then 4% thought the D was correct, and you don't have the ×1 because you're talking about the karyotype format. So that's the last slide. We've got a few minutes. If there are any questions, I'm sure Jean and I would be happy to answer them. Just to acknowledge the standing committee for all the work in putting this nomenclature together, the Genome Mapping Consortium, who gave us tremendous insight and feedback on the proposals that we had, and also gave us some examples, and to GenQA for letting me share some of their data.
I've answered several questions that were in the Q&A. So
right.
the attendees can see the answers to those questions. I think I've answered all of them. But if I haven't, please let us know.
Yeah,
There's a few.
Thank you very much for answering. You've answered a lot of them. I just want to, I'm not sure that everybody can see, so I'm trying to pick out the best one. What would be the best one that you thought was a great question? I'm kind of reading over.
Well, there were lots of good questions.
I know.
So somebody asked, when would you not use the tilde? And that's really when there isn't any ambiguity about the position of those DNA motifs that are involved. How do you describe a three-way translocation? I just copied in the example because I thought people would like to have it, but it is, I think, in the
book.
Oh, in the genome mapping chapter.
Yeah.
Somebody asked if the examples that we gave for using the tilde, all of them were for karyotype, not array-like nomenclature. So, but you can use it for both. Like you can use the tilde anytime you have any uncertainty. And then somebody asked if several structural abnormalities are detected by OGM and only one chromosome, should you always assume all of them happen on one homolog? I would say no. Or should we take a look and see if there's any overlap between the abnormalities? And I just said it's good practice to take a look for overlaps. Those things are really hard to sort out. Somebody complained about the quality of the physical copy of the book, and so we'll give that feedback to the publisher. Pages coming loose, I guess.
Also, write directly to the publisher. They may possibly give you another copy, because I did that and I got another copy.
Oh, excellent. Do we italicize the names of the gene in the nomenclature? No, just like fish, the names of the genes, although, you know, if you're writing a manuscript or something, the gene names should be italicized, but in the nomenclature, there's no italics for the gene names, just like fish. When do you give the VAF? It's an optional thing, but it might be clinically relevant, so I would include it if you think it's of value. And then somebody asked in the slide with the 22q and 17q deletions, how would you write it if the breakpoints span more than one cytoband? So just like microarray, if you have more than one cytoband that's involved, if you're using that array format, you list both of the start and the end of that copy number variant, and just start with the one that's closest to Peter. So you list both cytobands at the extent of the
Wonderful. Thank you so much. I'm gonna go ahead and say a big thank you. We will have these available as PDFs. I know there was a couple questions on that, but I do want to thank you guys for your wonderful job on explaining the updates. It was much needed. Just want a reminder, next week we actually have a MyeloMatch. MyeloMatch is the new clinical trial through the NIH. And Dr. Gerald Radish is going to give a presentation on that. Finally, remember that early registration ends tomorrow for the CGC annual meeting. So please attend and please send in your registration.